to just leave on for just a minute, and I'll give you the QR code on the next slide in order to make sure it was big enough for everybody. And then I will also show this at the end of the presentation so that everybody can get this. I know how important stuff, important information. so I'll make sure we have that, and I'll leave that up at the end. So thank you very much for having me. As Madeline mentioned, I did my residency at VCU, and surprisingly, I did my residency in community pharmacy. I never thought that I would end up talking to anybody about pharmacogenetics, but it's surprisingly been a very logical transition. And so just to give you a little bit about my background, because I started in that kind of ambulatory care community pharmacy space, and really focused a little bit over the years then on drug information. And that's the primary care space is a really active space for pharmacogenetics because of the number of drugs that overlap, and we'll talk about that in a little bit. But the biggest thing, and I think it's important, I think this is a tenet from what I have learned and the people who I know at VCU, I think the biggest thread that is common to both areas is kind of an entrepreneurial or innovative kind of endeavor or spirit. And so in the same way that, 20 years ago, we were giving vaccines, influenza vaccine for the first time at UCROPS Pharmacy in Richmond, and really trying to build a business model and a model for practice for changing the way that we do things in pharmacy. Pharmacogenetics is very much the same. A lot of what I do is very similar. It's just kind of trying to build models for practice that will work and be sustainable and efficient, whether that's with pharmacogenetics or influenza or diabetes. So it's been a really interesting kind of journey, and I'm glad to have kind of seen all the different areas of practice that I've been able to see. And so we'll start with just a little bit of background on pharmacogenetics. And let's see, here we go. And of course, we know that this is our way that we can identify what patients might be good responders, poor responders, or not responders, or at risk for toxicity based on the genetics of their drug metabolizing enzymes primarily, and also their receptors. And CYP2C19 and CYP2D6 are the two that we see most often. And this is really over the years, pharmacogenomics started out as more of kind of a science, a research science, where we were focusing on discovering genetic variants that influence drug response. Over time, we've continued to develop the evidence base and tools for the clinical use to really focus on implementation and then documenting that impact of clinical outcomes. And so I think a lot of people for many years still thought of pharmacogenetics in this drug discovery space, and there's certainly plenty going on, and there's certainly plenty still there, but we definitely have moved more into these last three bullets over the past 10 to 15 years, as we have been able to move more of that into the clinical space. One of the things that I think is very interesting is that pharmacists have and will continue to play very important roles in each of these steps. And it makes sense if you think about the role of the pharmacist as kind of our medication expert and leader, but also in the medication safety realm. And if you think pharmacokinetics, pharmacodynamics, there's a kind of a huge long history of pharmacists having a leadership role in similar types of services. And we see in the clinical space that our pharmacogenetics services look very similar to those types of services. We also have a lot more data. So the Clinical Pharmacogenetics Implementation Consortium is the group that provides guidelines for different gene-drug pairs. And you can see here are the ones that they have published. And so we have a tremendous amount of clinical guidance that we didn't have 10 or 15 years ago. Interestingly, I think that on every one of these guidelines, each one listed as a separate guideline, in some more than one, but each one has a pharmacist as a lead or a primary author. And so it really is not just even this theoretical space that pharmacists are really, kind of poised and well kind of qualified to take the lead. We see that really in practice. Most of our increasingly our pharmacogenetic programs within health systems are led primarily by the pharmacy presence. And a lot of the guidelines and national initiatives and international initiatives are also led by pharmacists. So our program started, the title of this program was kind of the last 10 years or last decade. That's about how long I've been working with our precision medicine program. And it started kind of in June 25th of 2012. And you can see there in that photo, that's the news release from that start. And that's our Julie Johnson, who is now our dean and the head of our pharmacogenetics laboratory and research program for many years. And she, at this time, felt like taking this into the clinical space would be a very logical next step. And so she's seen there with David Anderson, the head of interventional cardiology at the time at our health system. And they really were going forward based on the link between genetics and CYP2C19 variability and clopidogrel, specifically with outcomes after invasive management or percutaneous cardiovascular intervention. And this was kind of one of the first pilots that we did a one-year pilot there to just identify if we were going to be able, so is it even feasible to integrate pharmacogenetics within that space, and specifically in that subpopulation of cath lab patients or patients undergoing PCI. And so we did CYP2C19 genotyping was added as a standard of care for patients in the cath lab. It was pre-selected on order sets, and then all of those genotype results were moved to the EHR. And so in that first 12-month period, we genotyped over 1,000 patients clinically, and that was kind of our start and how we kind of, begin to learn our kind of how to implement lessons. And really with pharmacogenetics, you almost have to do it to learn how to do it. It's kind of what we've seen over the years. It's a little bit different everywhere, every setting, every gene drug pair. And so, so it really was the start of a 10-year plus learning process. Over time, we've added to that, and you can see this is just kind of a brief timeline, starting on the left-hand side with that CYP2C19 clopidogrel implementation there that's circled in red, and then kind of moving over the years, both on the educational, the research, and the practice side, where we added in our first PGY2 residency. We now have a resident and a clinical fellow, and we've had graduated residents since that 2012 year that have gone all over the nation to lead pharmacogenomics programs. And then over the years, the way that we kind of implemented, and like I mentioned, was kind of by, at least at first, by gene drug pair. So we said, let's start with CYP2C19 and clopidogrel and the small cardiovascular population, and we'll see if we can make that work. And then we kind of began to add on to that over the years. And what we've found now is that as we've moved on and kind of built the infrastructure, it's much easier to expand into different environments. And so now we really are threaded throughout the health system and have 2 outpatient clinics, consult clinics, where patients can get a panel-based or an individual gene drug test. And so over the years, that has been funded by a number of different grants, including, as we mentioned earlier, the Ignite 1 and Ignite 2 networks funded by the NHGRI, and then an NIH Genomic Medicine Training Grant Award. And education is a big part of all of everything that we're doing across the board from both the clinical and research side. So just like I mentioned on that previous slide, we kind of have progressed over time, and we have really at this point about 3 different categories of services that we offer within our health system. We do genotyping throughout pretty much every setting, so inpatient, outpatient, acute, chronic, kind of depending on which one is needed. And we try to keep the general procedures threaded, and we'll talk specifically about that throughout the health system for consistency. And one of the things that I emphasize, I think, is really scalability and being very practical and pragmatic with everything that we do. And so we have similar kind of the engine in place for decision support, provider education, communication, reimbursement, monitoring, so that we can kind of keep all those things the same as we build these different services and different settings within the health system. It makes it much more likely to be sustainable in the long run. It also helps us a lot from a scalability perspective. And it's a lot, when I first started, I had been primarily in an ambulatory care environment in an academic setting. And my clinical experience, the model that I was used to working under was you have a pharmacist who goes into the clinic, and if the pharmacist isn't there, then the service goes away. And so, it was really very innovative of Dr. Johnson. She said, no, we can't do that. We can't put a pharmacist in every clinic all over the United States and make this happen. We have to be able to build a system that will support pharmacogenetic testing. And the pharmacist's role is to help lead that and move practice forward, but it can't be dependent on the presence of one person. And so, so some of our services do involve direct pharmacist contact, but we do try very hard to kind of change the practice. So it's kind of an implementation science perspective rather than just kind of coming in and saying, okay, we're going to do this, and then when we leave, it goes away. So right now we have within our health system, it's the UF Health at the University of Florida, you can order single test. We work in-house with our pathology lab to develop and validate the test internally. We also work with some patients who get external pharmacogenomic testing, but you can order primarily 2D6, 2C19, or TPMT as single test, or we have a panel, it's called Gator PGX panel, that has the 2C9, SLCO1B1, and the genes listed there on the right side of that bottom half of that slide. I will tell you clinically, we do most of our genotyping and most of our recommendations, because we do consult notes and recommendations for every genotype that's ordered either on the inpatient or on the outpatient side within the health system. And most of those are for more of our chronic drugs. So our SSRIs, PPIs, clopidogrel, still we get a lot of clopidogrel orders, and more so than warfarin or simvastatin, or we do a lot of thiopurines also, more so in the GI space than in the pediatric ALL space, even though that's kind of where that azathioprine and 6-mercaptopurine are often thought of being used in. So just to kind of give you a little short journey of the life cycle of a pharmacogenetic test. And so once that order is placed, then the results are returned into the electronic health record. You can see there's that we use Epic as our electronic health record, and I'll show you a couple more kind of just FYI to show you what this looks like in the electronic health record system. But you can see that the gene that's tested in the consult note is placed, is also marked on here. And then we do a traditional consult note that's dropped into Epic. I think it's also sent via Epic in basket alert to the provider. And those are structured kind of like a typical SOAP note, but really just focusing in on the pharmacogenetic test results and recommendations based on patients' current therapy and potential therapy based on their diagnoses. And that test result is available. One of the biggest challenges with pharmacogenetic testing in the electronic health record is that it's not always easy or consistent to find where test results are, and that's something that's definitely we have struggled with over the years too within Epic, just due to capabilities. But the test results are in Epic, and they are a lifetime test result. The problem is there's not a lifetime spot yet for test results, and so they are sometimes a little bit harder to find, but they do carry with patients. When we build our clinical decision support, and I'll show you an example on the next page, the clinical decision support actually fires off of just the presence of a genotype. So even if you did, for example, someone within that pilot, and this is starting to happen more and more often now, in 2012, 2013, and did a CYP2C19 test for clopidogrel, and they came back to the hospital 10 years later and got, for example, a proton pump inhibitor that also had supporting clinical decision support alerts built into Epic. It would fire based on the lifetime presence of that genotype. And so the rules for our CDS are based on the presence of the drug and the genotype, no matter when that genotype is ordered. And you can see our best practice advisories, our clinical decision support follows a general formula here, where we give the option to remove the order and then to apply a new order. And then we try to include information about acknowledge or why you did not or did actually take this action or not accept the BPA. We'll talk a little bit more about kind of how to keep this systematic. One of the things that we really try to do again is have a very kind of structured layout for all of our CDS so that we can kind of plug and chug a little bit with different drugs to make it a little bit easier, because we do have such a high volume at this point. So just to give you an idea of where we have kind of come from 2011, 2012 now, you can see that we've genotyped nearly 6,000 patients. I think probably we've surpassed 6,000. Well, by now, I think this is a little bit old data. The age is about 57, primarily Caucasian patients, and primarily or a higher percentage of males. It kind of age-wise, it is primarily an adult population, but we do a lot of pediatrics, again, mostly with that TPMT GI side. One of the things that I wanted to point out, we have the kind of the ability, and one of the, I guess when we get to the lessons learned in just a little bit, one of the lessons learned is kind of tracking, thinking about the data. If you're starting a pharmacogenetics program in your institution, to really think about the data that you're tracking as you go forward, because you can learn a lot from it. And because we're so early in the implementation space, there is a lot to learn, tremendous amount. And so still, even just from how do services work. So just in the past few years, we've been able to set up a REDCap database, and a lot of our data are then automatically withdrawn from the clinical side, and then we can just run reports over time. And this has been very helpful. But one of the interesting things, then this just shows the number of genotypes per year, and a lot of this is driven by, for example, a research project. So I mentioned we have research and clinical implementations like side by side within a single clinic. And so a lot of this will kind of spike by the research funding or research projects that happen. But one of the things that we're really excited to see, and it doesn't seem like a lot on here, but when we started out, in 2011, before we did that first CYP2C19 clopidogrel, there were 21 genotypes ordered within the health system, just purely clinical organic, and we kind of think of these as would have happened, no matter what. We don't do anything to make these occur. Versus now, that slowly we're beginning to see that trend up to where a larger percentage of our genotype orders overall are clinical organic orders versus those that are in a research environment. And those 2020 numbers, I think that's about 6 months into 2020. And so I don't actually know where we ended up at the end of that, but we're definitely seeing more clinical use. Now there's a spike here. That was that first clopidogrel project that we did, those clinical genotypes. But even very specifically, you can really see that, and I don't know if I have the data for this, but even if we just look at CYP2C19 alone, it also really has driven up, and that kind of supports, Dr. Johnson's initial, approach of saying, let's change the practice, not let's drop a pharmacist in and let the pharmacist, do their job and then leave. We see system-wide increased ordering of things like CYP2C19 for appropriate use. This is just a little bit more information. I think that probably the top graph is probably the one that's the most revealing. So this is any service that ordered over 500 genotypes during that time. So to kind of give you an idea of where our orders are coming from, they are primarily in cardiology here. So about 3,700 were in cardiology, 500 in GI. I mentioned that we do a lot of TPMT, and then also CYP2C19 for proton pump inhibitors with our GI outpatient clinic. And then also in primary care, where we do much more 2D6 and 2C19 testing for pain, depression, and for to guide SSRI therapy. So I'll switch gears a little bit. I am watching the chat. If you guys have a question or want to chat anything, and I am kind of trying to keep an eye on that at the same time. So feel free to use that if you want to. I will say though, I don't have a T-shirt for anybody. So I'm watching this and I'm thinking, wait, there's T-shirts in the chat. Okay, so no, I don't have any T-shirts, but I can answer questions if you have them. So lessons learned, and I wanted to just kind of focus on what have we learned over the last 10 years that, and not just, about pharmacogenetics implementation for, on a kind of one-by-one, one-on-one scale, but how have we made this work throughout the entire health system? Because we really do have a program now that's integrated throughout all areas of the health system in a lot of different practice models. I lead a team of pharmacists. We have anywhere between 1.5 FTE for our pharmacists. We have a fellow, a clinical fellow. We also have research fellows, but we do have one that's purely clinical. We have a PGY2 resident at any time, and then we have at least 2 students working with us. And so we've kind of grown a robust team, and we work with a number of different physicians throughout the health system. And so, what have we learned over time with this? And I would say one of the things that I think has been really revealing to us is the importance of kind of consistency with the service. And so, over this 10-year period, we really weren't starting out, sometimes a project like this grows as, kind of grows and becomes and changes over time, versus we didn't start out 10 years ago to say, this is exactly what we want our clinical pharmacogenetics service to look like. And so we had a period about 2018, 2017, where our policy was to return consult notes, not on every single genotype ordered, but just when they were requested. And so, we would, for example, so any genotype might be ordered. We know it's ordered because we get an in-basket alert when any genotype is ordered, but we only did a consult note if somebody said, can you do a consult note for it? And I was really surprised when we went back and looked at the data, because we switched in July of 2019. we said, you know what, we're going to do a consult note for every test that's ordered, no matter whether they request it or not. And we're going to look at this as an opportunity for education and for awareness about the program and helping people to utilize pharmacogenetic testing the correct and appropriate way. And what we found is that if we compare year 1 to year 2, so this is that first year where we were only doing test or consult notes on request, to the second year, we had a dramatic increase in the number of tests ordered. And so from 191 to 310, about the same services, but what we saw was that of the tests that were ordered, and we look at those and say, okay, did this person follow or not follow the recommendation of the pharmacist? And so this was a general, like a consult note, like any other one, the pharmacist says, based on the genotype, you should do this or you should not do that. And what we found was due to the increased number of medication of genotypes ordered, consult notes placed, a much larger number of recommendations were accepted and being able to implement it into the hospital. And so the impact that we were having as a system within that system as a pharmacogenetic service was much greater when we were actually really using that opportunity, even when there wasn't requested to provide a consult note and offer our help and services to those patients. And so one of the things that we've learned is really the importance of being consistent and consistently having that clinical voice within the space of pharmacogenetics. One of the other, I think, really important things that we've learned is, and I just kind of alluded to it earlier, but it's more towards scalability. And there's a lot of moving parts to a pharmacogenetic service. There's, and I kind of showed this on the umbrella slide earlier, there's patient education, provider education, there's lab report language, interpreting lab reports, there's clinical decision support. There's a lot of pieces that have to be built for every time you do another gene drug pair. If you work with a commercial lab externally, a lot of those pieces are built for you by that company. But even then, you still have to be aware of what the pieces are. And so we had a period between about 2017 to 2018 where we just ramped up when we went to that panel. So instead of doing just those single gene drug pair tests, we went to that panel testing, and we needed to ramp up really quickly. And so what we did was we actually kind of thought about all the things that we had to create. So for each pharmacogenetic test that was going to be ordered or resulted in our health system, we needed to have a lab report. We needed to have the clinical decision support. There needed to be a note, something that went to the provider that gave a recommendation, and we needed patient education. And so we went through each one of these and said, what are the common pieces of information that are going to be essentially the same? So phenotype, we can give the same phenotype to every single person and every single, pathology report, decision support, but what we offer or what we, the recommendation we make is going to be different. So for a pathology report or for patient education, we're not necessarily going to make a drug therapy recommendation, but for the CDS and for the provider report, we will. And so we just kind of broke this down in a systematic way to say, where are we going to need the different types of information at which step? And that then gave us the opportunity to say, okay, for every new clinical decision support we make, these are the things that we need. And so it kind of gave us that opportunity to go year to year very consistently and just kind of build almost policies and procedures or a process for how do you build clinical decision support. So we know now we need these. It also helps us to keep things consistent within the health system so that we're internally consistent with our own CDS builds, because we do have so many now. And so what we're able to do is kind of based on that and based on knowing what we are going to need at a certain time, then we built matrices or matrices that gave us the opportunity to say, okay, for this diplotype of CYP2D6, this phenotype is going to show. It'll be triggered by these medications. So codeine and a CYP2D6 ultra-rapid metabolizer is going to generate this problem statement, and this problem statement will be used for the consult note and for the clinical decision support and this recommendation. And so we standardize these pieces so that way everything that's built within the health system is consistent internally, and it also helps us when we have to change something, we know what to change. And so it's much more efficient than trying to kind of look at each one individually. And I think this is one of the strengths of having a systematic approach within a health system, especially, is the opportunity to think about things like in this manner to really make sure that everything that's happening within your institution is happening the same way and the same kind of based on the same level of evidence. This does get a little bit more complex, and I won't I won't spend a ton of time on this because it's a little bit, it's complex to even look at it. But if you think about what I just showed you, it was a 2D6 diplotype, a phenotype, and a med, really one medication, because you're not going to be on all four of those at the same time. So this is truly a gene drug pair. Okay, you have one gene, one drug. As we get to learn more about pharmacogenetics and about drugs and what are influenced by certain genes, we know that that's not always the case. We have two or more genes that affect a single drug, and we're going to really see that much more often. And so it's really easy when we're starting out with a single gene drug pair, it's pretty easy to kind of just lay out, a little outline of, okay, if this, then that, to drive what your decision support or your decision making is going to be within the health system. It gets really complex when you look at, for example, a 2C19, so an SSRI, an antidepressant, is affected by both cytochrome P452C19 and 2D6 variability. And so you have to kind of think through, well, what if the drug is ordered and we don't have 2C19 results, then, or we don't have 2D6 results, we only have 2C19. So then it's going to follow this pathway. What if we have both? What if there's no genotype? What if, there's a lot of different what ifs, and this becomes very complicated. And so we also worked out a system for addressing kind of this complexity of multiple gene drug pairs, which took us honestly a little while. But once we worked through this, then we can just drop new genes and drugs into this. That makes it real simple going forward. This is just kind of the same, what that would look like in the matrix example for both of the CYP2D6 and 2C19 phenotypes, where you're going to have a little bit more complex logic, but it gives you where it kind of shows you if there, for example, I'm sorry about that, hold on a second, a CYP2D6 ultra rapid metabolizer, then they'll have this. This is what we need to do for them for both their problem statement and their BPA text. So again, this is kind of just closing that loop. That information then is put directly into the BPA. And so what I showed you on this page, a problem statement in the BPA text, then that goes into certain specific spots on the BPA. And again, just helps to make that very clean and very systematic throughout the program. And building this out earlier, I think has been a big lesson that we've learned over the last decade. Another important lesson, and I alluded to this earlier, is really trying to look at what data are you collecting over the years, and what data are you able to collect within your system. Pharmacogenetic implementation is still very, very new, and so we don't know some basic things that we do know about a lot of other areas, that we don't necessarily need to know, what does someone do when they see, a pharmacokinetic level, or what are they even going to do with that? Are they going to act on that or not? We have a much better feel for a lot of other kind of much more implemented, services. And so it's a good idea, if you can, to collect any data that you can and watch it and learn from it, because you really can. And so the thing that's most recent that we are, I think, most excited about in this space is our BPA data. So we weren't able to get really good data out of our best practice advisory firings within Epic for a very long time, but we just started being able to do that. And so now we're able to kind of see, okay, within our system, with these BPAs, because our BPAs, we build these and the clinical decision support kind of runs automatically in the background, we're still able to collect data on what's firing and what actions that clinicians are taking as a result of that BPA firing. And so we're continuing to monitor and collect more data in this way. Last thing that I will mention from a kind of a lessons learned perspective is really thinking about where you're looking at for pharmacogenomics and kind of where you're placing your energy and your efforts. And I think that a lot of times people, like I mentioned earlier, you don't necessarily think about primary care in pharmacogenomics as much as you might think, for example, about cytopurine methyltransferase testing and pediatric ALL. But we really have grown and been working a lot in the space of primary care, both from a kind of an implementation, so a traditional implementation standpoint of is this feasible, can we make it work, but also in building a business model and a reimbursement model. And so we worked a lot with our primary care clinics, and I think we have two, both with family medicine and internal medicine. We have multiple, probably 2 or 3 actual physical clinics where we see patients, and I have the structure here. You can see it's kind of a traditional, primary care structure where you have there's a 2-visit, follows a 2-visit structure, with the first visit kind of being education, medication reconciliation, and identifying, is this patient, a candidate for pharmacogenetic testing? And then that test is taken, the sample and the result is back into the EPIC, into the electronic health record, and we have a second visit where we can then do education and counseling. And we have really tried to set this up ideally in such a way that would kind of maximize kind of the reimbursement and the compensation for the pharmacist too, knowing that it's not just the pharmacy, not just the pharmacogenetics test that needs to be reimbursed, but it's also the pharmacist's time, just as with any service that's going to be run by a pharmacist. And so we really try to be very conscious about looking at different ways to approach our clinical care and that reimbursement, not just for the test, but also for that pharmacist. And we do that a lot of ways by kind of building on the ambulatory care and other payment structures to kind of think what are ways that we can bill for and are appropriate to bill for, given the current climate and kind of what's going on with this. This is just another image of that visit that I just showed you. You can kind of see here in that where we have 2 visits, we have 2 potentially billable visits, both the first and the second. And we do currently bill for both of those in the clinics that utilize our 2-visit model. We also are testing a 1-visit model just to see if, does that level of efficiency, interaction with the patient, is that overall more effective and work better in the long run. The other thing that I wanted to just point out is that we talk a lot in pharmacogenetics about evidence and the importance of using your clinical evidence and evidence-based testing. And that's that's a key. We really have to know, and I showed you the slide earlier with the Clinical Pharmacogenetics Implementation Consortium guidelines. And that's really, we're looking at that strong threshold of evidence. But another thing that we've learned really is to kind of look at the practice-based evidence before we begin a specific pharmacogenomic implementation. And what I mean by that is really we're not going to go into a clinic even if there's a desire. So if we get a request from a provider, say a psychiatric provider, that they're looking for pharmacogenetic testing, the first thing we're going to do is say, is it going to work in your clinic? And so this is just kind of 2 examples of some of the background data that we might pull before we actually even start to think about implementing, especially in an outpatient setting, where we'll look at patient breakdown, condition by condition and payer. And so looking at, are there patients in this clinic who would benefit from pharmacogenetic testing that are, say, being treated chronically with antidepressants or pain medications? And of those, which ones are those that we know are associated with a high level of evidence from a kind of an evidence-based perspective? And so we'll look at the specific drugs, whether codeine or tramadol, oxycodone, specific SSRIs or PPIs, so that we can say, okay, in this particular, like you can see in table 2, in this particular scenario, out of all of your patients, 33%, so more than a third, were taking a medication that's associated with a high level of pharmacogenetic evidence. And so we feel comfortable that we can make an impact clinically within this space versus we don't want to go into a space and there's really no value. I think that's one of the big things that we have kind of taken home with this is that if we aren't making evidence-based decisions, or if we're doing implementations that don't actually, affect any patients, then it really devalues pharmacogenetics as a whole. And so we try not to just kind of do anything we can do, but to thoughtfully do what was going to be the most beneficial for the patients and for the health system overall, and doing that in a way that will also be sustainable and scalable. Oops. And I went forward. I should have gone too far. So I'll kind of wrap us up, and then I'd be happy to take any questions that anybody has. But what we've really found over this time period is that we can implement pharmacogenetics within a clinical setting. It does help to have initial research funding or some initial funding to start that up. But what we try to really do is use the research funding as a catalyst for our clinical practice change. We have definitely seen within our service that pharmacist leadership is really essential in making that the PGX services work and making them also work over time. Education is an important catalyst. I think we touched a little bit on education. And when I say that, I mean education of not just our providers, but the pharmacists, the workforce, your students, your residents, your fellows, and then also, providing that clinical training as an APPE space. I think it's not every school of pharmacy or health system can support a clinical rotation or ACPE for pharmacogenetics, but I think that having that available as soon as possible within an institution is really important, because so much of what we do in pharmacogenetics is kind of more of a hands-on learn. It's more of that innovation, entrepreneurial, hands-on. You really have to be an advocate for pharmacogenetic testing, and you really do need to be a leader and be able to kind of identify stakeholders and win them over, because there is still a level of kind of convincing people to some extent, or breaking down barriers with pharmacogenetics. And so that education is really just as important in a lot of ways as the science. If you have that really strong foundation of science, but you can't do anything with it because there's nobody knows what to do with it, then that's going to be a shame, and it's going to be a missed opportunity there. And then within our own health system, we do try to keep things as much as possible, consistent, scalable, efficient, so that they can move throughout the years. And I would say probably that's one of the biggest lessons learned over time, is doing things very systematically helps in the long run, even if it doesn't seem like it might matter very much at the very beginning. So before I take questions, I just want to say thank you to everything. This is a team effort, so a lot of people are involved with our program, and you can see they span from this is a multidisciplinary program, from physicians to pharmacists, pathology, administrative, research, and then here's a list of our previous PGY2 resident trainees. And I should have shown, I have a map where everybody is now, where everybody lives, and then the NIH grants and others that we have received funding for as a group. And but great group to work with, and this is a great team that has made a huge impact at this health system. And so, so I can take questions now, or anything else that anybody has. I'm happy to do that. And I have also the instructions for claiming credit, so I can go on to those if we need to. And so. So I'm going to go ahead and go on. I don't hear any questions or any, oh, I do see something in chat, so I'll leave this up. And the question that's in the chat is, were the pharmacogenetic test results shared with patients, and do patients' perspectives and attitudes have any effect on the implementation process? So that's an excellent question. We share tests on the inpatient side. They're considered to be kind of the part of the clinical, the same clinical process. So they're shared with patients in the same way on the inpatient side that a serum creatinine might be. In other words, it's in their laboratory file, and their clinician's going to use it to make a decision, but we won't necessarily, we don't come back just the same way we don't come back to a patient and say, here's your serum creatinine every day. We kind of, it's put in their file and it's in their medical record on the inpatient side. On the outpatient side, it is a little bit of a different process. We have an educational video that we show, and that's it's pushed into, so before patients have an appointment with the pharmacogenetics consult clinic, they receive an electronic message within the electronic health record that links them to a video. It's an educational video about pharmacogenetics, and then afterwards they receive a print copy, and then also their results are in MyChart, which is the kind of patient-facing portal for Epic. And so they receive those both in writing, they're counseled on them, and then they also receive them electronically. And we do research. And I apologize, I just was looking at these data last week on patient satisfaction, and especially early on, things like willingness to pay, changing perceptions. We've done a lot of research on kind of changing perceptions with providers also. If we educate providers, are they more or less likely to order a test? If they undergo genotyping themselves, they're more likely to participate in genotyping. And so we do look at satisfaction, and that definitely does impact our services. Each one of these data points, whether it's from the patients, the providers, or the health system overall, is kind of, we're constantly working to feed that back in to change and kind of grow what we do over time. The next question is, one argument I've heard against implementing PGX is the turnaround time for testing is too long. So great question. How have you kept the turnaround times fast? Do we have a lab in the hospital, or do you use outside providers? So this is really an issue when you're talking about your acute setting. And so I mentioned that a lot of our testing has moved into the outpatient setting, and it's a lot of driven because of this. So in the acute setting, a great example would be CYP2C19 and clopidogrel, where you really need to have, if you're going to make the most meaningful change, the most meaningful time that you would change from, for example, clopidogrel to an alternative antiplatelet agent would be immediately after the test, or immediately after the PCI. And so the best time to have that test result is going to be before that procedure is performed. Unfortunately, the payment or reimbursement for testing beforehand is not very good at all, because then it's considered a preemptive test, and so there's nothing to kind of structure that billing around. And there's only one test that you can get a quick turnaround. So there is a test called the Spartan test, but it has to be performed, at least last time I've seen it used, it has to be performed in the pathology lab. And so it works well if you have like a PCI lab and a pathology lab right next door to each other, because then they can run the test back and forth. But that's kind of like the only way that we've been able to get like a test, a CYP2C9 test result within an hour, for a PCI patient. That's where it really does make a difference, because that it gets tricky if you can't do that. And so for a lot of our PCI patients, they do a follow-up. And so within a couple of days, they'll follow up, and we try to get that as quick as possible. But that's kind of the worst case example of, you're right, it's if you can't have it very early on, then it really decreases the value of that test result. Our typical turnaround is 2 to 3 days. We do work with an in-house laboratory with pathology in our own group, but we also do have test results from outside. The test turnaround time isn't as much of a big deal now as it was probably in the very start of working in this space, because so much of what we started with in pharmacogenetics was the specialty care and inpatient care. So cardiology, oncology, very specialized for these groups of patients. And so they were much more likely to be acute, whereas now we see this kind of recognition that a lot of the drugs that are affected by pharmacogenetic variability, especially 2D6 and 2C19, are chronically used drugs. So SSRIs, proton pump inhibitors, where waiting for a week or two for a test result isn't the end of the world. If you're looking to, for example, either identify the right SSRI or the best, not the best, but identify an SSRI to start with based on pharmacogenetics, it's not bad to change a week into therapy. It's not, you can kind of move around a little bit more flexibly. And so we don't see this as much of an issue now as we have done in the past, but it's because a lot of the pharmacogenetic testing is shifting to that outpatient ambulatory care environment with drugs that aren't necessarily acutely changed or started. And the last question I see is, are the Gator PGX panels done internally? Yes, all of our testing is done internally. And our group, it's kind of this is unusual, but our group, because of the amount of research that is done within our program, we really work with pathology to validate and to set up those panels specifically. So we actually meet on a weekly basis and kind of go through. And so if something comes out, something happens, and there's a new test result or a new data on, say, CYP2C6*10, then we'll do an evidence analysis, and we can advise pathology on whether they would make a change. So we're very heavily embedded in that kind of lab validation and development process, which is not, I don't think every system can support that or that level of interactivity, but we're very much looking at that and changing those so that we can then base all of our, everything's then based around one system. So we can kind of customize the pathology report with the clinical decision support and all of those. Cost effectiveness of our model, quick question about that. So if our tests are research funded, a lot of times we're actually studying implementation. And so the Ignite research funded tests are the Ignite network that we're funded through under the National Human Genome Research Institute. The purpose of the network is not just to see what happens from an outcomes perspective, but also to see what models are feasibly implementable. And so some of the time we are funded by research to study how to implement. And so many of those implementations have been funded by research, but as we go forward, everything we do clinically, we try to be self-supporting. And so, for example, our pharmacogenetics consult clinic, we bill test for patients through their insurance. We have a practice agreement in place with the providers, and we work in conjunction with the providers so that they're able to be a billable visit. I don't think we're at the space where we support ourselves. I don't know if any pharmacy service is there, but we are very cognizant of it, and we do try to we really can't take up space in a clinic unless we are, cost effective, and unless we're at least, kind of cost neutral. And so before we do any of our kind of outpatient implementations, we do look very carefully at the business model and the administrative costs, RVUs that are generated, the drugs, so that we can make sure that we are cost effective, if not cost neutral, for that space. And then, do insurance companies cover the test fee and provide alternative treatment immediately if needed? The insurance company coverage varies. We talk to patients ahead of time. What we have done is work with our pathology lab to say, here's what you'll be charged on a cash basis if your insurance doesn't pay. And then if your insurance does pay, it can be anything less than that. So patients are never surprised. They're not going to get a huge, bill. It's going to be a couple $100s. But we don't know. We've done some data analysis on insurance. So we do track and monitor what insurance companies pay for which test. It is completely and totally random. It's like there's actually no rhyme or reason to it whatsoever. And where we tried, we wanted there to be rhyme or reason to it. And so we watched the data very carefully, but it's really hard to predict when an insurance company is going to pay for a test. And so we just kind of treat everyone like they're not going to. And usually the insurance company doesn't provide any recommendations. They just provide the test result. And I'm sorry, not the insurance company, but the lab just provides the test result. And the insurance company is just a claim that's processed just like any other claim. And so they don't provide any recommendations, which is why I think having that pharmacist voice to really weigh in there is really helpful in this space, because there is a lot of education and kind of ambiguity about what to do with those test results. And so these are great questions. So I think that's all. Let me go on to the QR code so you guys have that and can scan that and hopefully have everything that you need. And so, and I'll wait a minute and see if any more questions come in, or if anybody else thinks anything, just let me know.
Hi, Kristin, this is Yusuf. Thank you so much. Hi, thanks so much for actually talking with us. And I appreciate you sharing your pearls of wisdom from UF to Richmond, Virginia. So thank you for that.
Yeah, thank you. Thanks for the opportunity.
Yeah, no, it's great. I know this definitely was not the purpose of your talk, but can you share a little bit about the outcomes? Obviously, the clinical outcomes. Obviously, Julie and Larissa and yourself, you published on this. But can you give us just a snapshot, basically, how the implementation led to better clinical outcomes? So, and how many lives were saved, and how many hospitalizations were prevented? Can you just, briefly talk about that?
I can, and I apologize that I didn't show any of those data. And we do have data, and I'll try to actually, I don't want to leave my but I'd be happy to send. We have a number of papers in this area. A lot of times what we see is, of course, our gold standard for evidence in that outcome space is our randomized controlled trial, which is very difficult in pharmacogenetics. And so when we started out for that, for example, that CYP2C19 testing, we were looking at feasibility. We began then to look retrospectively at outcomes, primarily in pain patients, in CYP2C19 and clopidogrel, where we were able to pull data with. So for example, with that particular gene drug pair, even with the number of genotypes that we do, it's still not enough to be able to show a statistical effect on outcomes. And so for CYP2C19 and clopidogrel, we worked with a number of other institutions, essentially anyone that was kind of doing pharmacogenetic testing clinically for CYP2C19 and clopidogrel, and pooled our data nationally and showed a decrease in major adverse cardiovascular events as a composite outcome. And so we have definitely seen a decrease in cardiovascular. We've also seen increased pain control in our CYP2D6 guided testing for both post-op and other areas using either codeine or tramadol and other things. And I'm trying to think of where else, that we have plenty of data. It's primarily in those two spaces. One of the questions you asked specifically was how many lives were saved, how many dollars were saved. It's a little bit harder to quantify as a single institution. We do have some collaborative publications for kind of looking at economic and cost effectiveness and utility from more of a research perspective. And so I apologize for not sharing some of those things, but we definitely do collect those data at the same time and have data specifically in that CYP2D6 opioid space and CYP2C19 clopidogrel, as long as from an outcomes perspective, as well as a lot of other kind of more earlier data for many gene drug pairs.
Great. Thank you so much. That's great. Thank you.
Yeah.
All right. Any last questions? This was really terrific. Thank you so much for taking time today out of your busy schedule, and several direct messages from folks saying how much they enjoyed the talk. So
Great.
Great job. And at some point, maybe we'll have you back on campus.
I'd love that. I'd love that. Thank you guys for the opportunity. I appreciate it. And so have a wonderful day.
You too. Take care.
All right.
Thank you, everyone.
Thanks, everyone.